The perennial sort of yin and yang of the two NaV1.6 inhibitor programs. So that's 562 here. So we're really grateful for the opportunity to come back and share the story again with the audience, but also some of the updates that we've been able to make with the clinical data, which we'll show in the slides, but also sort of a general update on the Praxis pipeline and where we think we're going. And all right, so at Praxis, we were founded in the genetics of epilepsy. So we've had some segues along the way, but in the spring, we really doubled down and came back to the epilepsy. And it's one of our core pillars of the company, targets that are founded in the genetics of epilepsy, because I think, as we all know, those genetics really point us to places, nodes in the brain where we can really control excitation inhibition balance. And so this is a general view of the portfolio at Praxis. In the left column, you're seeing a lot of the discovery programs, PRAX-222, and then PRAX-20, 80, 90, and so on. All of these are they are precision programs going after genetics of rare epilepsy. In the middle, we've got other programs that are targeting epilepsy genes. It's a GABA PAM at the top and a T-type calcium channel blocker at the bottom, really, again, leveraging the genetics of epilepsy to drive where we think we can make a difference for patients. And then on the far right, that's what we're going to talk about today, PRAX-562 and PRAX-628. So these are both small molecule sodium channel inhibitors. It's very, their claim to fame, so to speak, is that they have a greater preference for persistent sodium current. This is something with 562 we published earlier this year, so we're really not going to belabor the points other than to say 562 is about 500 to 600-fold more potent than any of the other sodium channel blockers, so the carbamazepines, oxcarbazepines, cenobamates. With this, you have greater preference for persistent current. You have greater activity dependence of the molecule. So what we think we're doing is really targeting the hyperexcitable states. When the neuron starts to fire, which is what sodium channels do, we're able to quiet that activity down. When the neurons are not firing excessively, we don't engage the target. And so it's really a way to modulate and to drop pathophysiologic hyperexcitability. And that's really what we think we have 562. So it's not an isoform selective agent, as we heard earlier, but it is a biologically a pathophysiology selective agent. We think that's being read out here. So we have two slides of preclinical data. The first, this is a classic MES assay, so electrical stimulation, generalized seizure. You can see with increasing doses of 562 in this mouse model, in this mouse, these are wild-type mice, we have protection from MES evoked seizure. So it's not surprising with sodium channel blockers. On the right, we have SLMA. So this is tolerability. So this would be a spontaneous movement in the field, and it's a sodium channel blocker. So as you go higher, you're going to get reduction of movement. What's interesting, what's important about these data and important about 562, the Praxis programs, is the separation between those two. And so the efficacy on the left and the safety on the right happen with about a 16-fold window. And that's shown in the bottom, 16.2. And as we showed in the epilepsy paper earlier in the spring, it's 16-fold for 562, but this compares to 4-fold or 5-fold for lamotrigine and carbamazepine. So more of a standard sodium channel blocker profile. So we really think that this wider preclinical protective index or clinical tolerability measure is really derived from the fact that we have this preference for persistent current. So this normal current that happens at subthreshold potentials, but can be upregulated either by mutation of the sodium channel or regulated by signaling, phosphorylation and such. We really think that if you're able to selectively target this persistent current, you're able to blunt the hyperexcitability and really leave the normal properties of the membrane, normal properties of the channel intact. So we think that's what's driving this 16-fold. Now, this isn't just solely dependent on wild-type channels. These are data which we showed at AES in December. But we have two different models working with collaborators. These are both gain-of-function sodium channel models. On the left, it's the Q54 SCN2A model with Jennifer Kearney at University of Northwestern. These mice have spontaneous seizure, which we can block with increasing concentrations of, or increasing doses of 562. On the right, data for Manoj Patel's lab at UVA. This is the SCN8A gain-of-function mouse model. Again, it's the N1768D model. This model is kind of cool because it has this audiogenic evoked seizure. So you turn on a sound, the animal sees. We're able to completely eliminate that by pre-treatment of 562. And what's not shown on these slide, but what's important, both of these effects happened in the Q54, it happened fivefold lower exposures than what we needed for the MES assay. And for the SCN8A model, it happened about the same exposure that we needed in the MES assay. So we're seeing anti-convulsant activity for 562, both in wild type and in genetic models, all about the same plasma and brain exposures. So sort of summarizing this and moving quickly, we think we have selectivity, but we have selectivity for pathologic hyperexcitability. So we know sodium channel blockers are important. We know that they suffer from tolerability. We know that a lot of this is Cmax related, which you'll see some PK in a second. So it's really an on target. We think 562 is going to be a better type of sodium channel blocker, primarily due to this selectivity for persistent current. We have an unprecedented therapeutic window, preclinical protective index, and we're driving towards that for clinical data. And the interesting thing here at the bottom is what's termed a convenient auto titration regime with stable PK. So we'll get to that in 2 slides. These data are coming out of the MAD cohort. So this is 562-101. So we've completed a couple of phase 1 trials now, and this is just showing some of that data, really focusing here on treatment emergent AEs. And here, all of the AEs that were observed were moderate in severity. There were no drug-related serious AEs or severe AEs. There were no significant aberrations in clinical safety labs, ECGs. So, we're not seeing QT prolongation or anything like that. No increase in suicide ideation, and there was not a food effect of the molecule. So it seems to be fairly well tolerated, a really nice profile to move forward. And just noting that the things that you see here, like nausea, vertigo, those are typical CNS AEs or AEs of typical CNS active drugs, and we were either at the same level or even lower than what you typically see with those agents. So it's giving us a lot of confidence in moving this molecule forward. In terms of exposure, it's the second half. Where were we? And so it's kind of a busy slide, so we'll spend a minute or two here. So on the left, you're seeing day 1 exposure, so single dose. On the right, you're seeing the exposures after over 28 days, so multiple doses. And the horizontal lines that are red and blue, the red is really standing out. Not that it's a danger signal. I would not have put that as red on the slide. But that's where we want to be. That's the EC50 for the MES assay. So we think we need to maintain coverage above the EC50. The other bluish line is the EC90. So same assay, MES. So we think if we get to EC50, we're going to be good. If we get to EC90, even better. This will give us complete coverage of that preclinical model and of the 2 models, the SCN2A and SCN8A model. And what we see is even after 1 dose, the PK, we're able to get for the 90 mg and then 120 mg, we're able to get above the EC50. And as you dose, the exposure titrates up. So the same dosing, this is 90 mg over 28 days. This is what we're referring to as auto titration. So it's going up over time. It quickly goes above the EC50, then it goes above the EC90, and you see a narrowing of in this study, narrowing of the peak to trough ratio. So all of these are good signs in terms of how we feel about the PK for 562. So we think we are in the exposures we need to be. We think that with a profile like this, there is less risk of missing a dose because you should maintain coverage with this PK profile. So these data are giving us confidence in healthy volunteers, giving us confidence to move the molecule forward. That's really the last slide here. So this is slide 8. 562, it's really it's leading the way, but it's quickly being followed by 2 other programs. So by the end of the year, we hope to have 3 molecules in the clinic, all targeting epilepsy. So 562, we're going after SCN2A, SCN8A, DEE. It's a great place to play. Both of these are gain of function, as we heard earlier, which saved me a slide of background. We're also going into TSC. And so we hope to start those trials by the end of this year. PRAX-628's molecule, we talked just briefly publicly about. We won't talk about it here other than to say it is related to 562, and that molecule we intend to put into focal epilepsy. We're starting we nominated it last year. We're starting phase 1, hopefully by the end of this year. And PRAX-222, this is an ASO, so not a small molecule. Praxis is not just a small molecule company. So that's an ASO. It's designed to target and downregulate selectively the SCN2A mRNA. So again, going after SCN2A gain of function. We're hoping to initiate a seamless study by the second half of this year. So, and then you can see some different designations that we received from the FDA and the EMA. So in summary, Praxis is well positioned. We're really excited about the assets that we have in the clinic. We're excited about the preclinical pipeline, really doubling down on epilepsy. So we hope to see all of us for years to come. So thank you. Take any questions.